All right, hello everybody. It is 2:30. Welcome back to our joint SPNHC 2024 conference. This is session CTRB, Contributed Oral Presentations, History and Collections theme. And this year's meeting was made possible in part by the generosity of our sponsors, especially our diamond sponsors. We are grateful to them, and we are also grateful to OK Congre for helping us host a successful meeting this year. If you are attending virtually, please ask questions using the chat feature in Zoom. We will ask the presenter your question for you. For those of us in person, please try to use the microphone so that your question is captured for the online audience. And after the session, feel free to move the conversation online to the Oxford Abstracts portal. Now, as a final reminder, by attending our conference, either in person or virtually, you have agreed to abide by our code of conduct. Now, I'd like to introduce our first speaker this afternoon, Diego Tavares Vasquez, who is presenting the history and importance of the, I can't read my own handwriting, I'm so sorry, of the fern herbarium collections in Japan, with focus on the University of the Tokyo Herbarium.
Hello, everybody. My name is Diego Vasquez, and I work as a research assistant professor at the University of Tokyo and as the curator of the TI Herbarium of the University of Tokyo. I'm not a historian. I work with ferns and lycophytes taxonomy, but the herbarium at the Tokyo University is one of the oldest here in Japan. So working with history there is quite mandatory. I don't know much, but I'll try to tell you a bit of what I found out in this past year that I've been working there. Using the index herbarium, I produced this map. This is the location of herbaria in Japan. The dots on the map, they basically show the institutions. And the colors, they mean how old those institutions are. You can see that the darkest spots, they are concentrated in the Kanto region, in the southeast area. And that's basically because most of those most old institutions, they were created after the Meiji Restoration at the end of the 19th century, alongwise with universities and museums that were created in Japan. So within those, we have the TI Herbarium. The TI Herbarium of the University of Tokyo is divided into two buildings here in these pictures. It was founded in 1877, and it accounts actually to close to 2 million specimens, with main collections from Japan, Korea, China, Taiwan, and many other locations in East Asia. The thing is, this is a bit outdated information. This data from the Committee of the Japanese Classification Society from 2015 compares the numbers of specimens from different institutions, and I brought the biggest numbers compared to the TI University of Tokyo numbers. And you can see that for vascular plants, we have a considerable number of plants there. And not only that, a good part of that are ferns and lycophytes, which is the main topic that I study and I'll be talking today. But what happens is that the TI Herbarium is famously known here in Japan for the huge number of type specimens there that are not cataloged. We don't know where they are and how many we have in the institution yet. So I've been working with that in this past year. I'm trying to catalog and digitalize all of the type specimens in the institution. And at the moment, for ferns and lycophytes, I found more than 1,000 specimens, and that's close to everything that we have there. But for gymno and angiosperms, we have a long way to go. We have already 5,600 specimens, which compared to other institutions in Japan is already like more than almost threefold the numbers. So the collection is not only known for the huge number of type specimens, but also because key people that worked there and created this collection worked not only in Japan, but also in other places around here. So we have, for example, Takiya Kawakami and Bunzo Hayata that worked with the flora of Taiwan, Takenoshin Nakai, which worked with the flora of Korea, but also Indonesia and Ceylon. And in Japan, we have the founders of the School of Botany at the University of Tokyo, so Jinzo Matsumura, Ryokichi Yatabe, and then from there, Tomitaro Makino, which is famously known for identifying and naming several species. So all of them together, these numbers that I'm showing here for the taxa that were named by these people are only for ferns and lycophytes, but of course they work it with other groups of plants as well. And all of them, they work it not only for Japan, but for other countries around here, having a very impactful contribution to East Asian flora studies. However, as we don't know the numbers and location of the type specimens, we don't know much about their collections in depth. We don't have a database of that yet. And if we don't know that, actually people outside of Japan probably knows less than us. So that's actually a huge issue and a huge mission that we have at the Herbarium. And I've been trying as much as I can, work actively on ways to show the value of that collection and try to bring that information to more people to see. So I'm going to show 3 examples of things that we could achieve in the last year. So the first one is just typification work. So I worked with this colleague from abroad, and we used the theme for this publication was this Maraciaceae family of ferns. In specific, these 2 species, Angiopteris tandauensis and Angiopteris tonkinensis. The type specimens for this group are in the TI herbarium. And these are plants that were described for Vietnam by Bunzo Hayata and in 1917. So these plants are 100 years plus old. And there are many syntypes, many duplicates of these plants in the collection. So we have to work not only searching for all of the collections from that trip, but also discussing the taxonomical issues around these plants. So us, as a specialist in ferns, helped with that. And then we were able then to finally lead down to one of the lectotypes of this collection and publish a work on that. Hayata Bunzo's specimens are very interesting. Their construction are quite messy most of the times and full of information in the specimen vulture. So it's quite of a unique kind of collection that we have there. Another example, this one was a work that we did in, it was a conjoint work between the University of Tokyo and then the Tokyo Metropolitan University MAC Herbarium and the National Museum of Nature and Science TNS Herbarium. And we studied these several groups of plants that were described by Makino Tomitaro. And here I'm bringing the example of Asplenium antiquum, described in 1929. Makino Tomitaro described more than 2,000 species over his work, over his lifetime. For ferns, more than 90 taxa were named by him. But most of cases, he collected many duplicates. In some cases, we have up to 50 syntypes of the same species, which is quite of a job for us to gather everything. And they are usually scattered around different institutions, so they are not all in the same institution. So we found all of the specimens in these 3 institutions, and then after we did that, we were able to decide which was the type specimen to be selected as a lectotype for this group. The last example that I bring are a few of some unique collections that we have there. These collections, they are restricted to which kind of institution they are kept. Here I'm bringing 2 examples. One example from the left side is the holotype of Hysteopteris incisa variety ceramensis. This plant was collected from Ceram by Kato in 1983. And plants from Ceram, collections from Ceram are quite rare. We have a huge collection in TI. They're in a good state, but we don't have digital information about them, neither a list of them yet. So that's definitely something that needs urgently to be databased. On the right side is something that I found a few months ago, actually. It's a collection from Masayuki Takeuchi from 1960, from Brazil. I'm originally from Brazil, actually. And this plant's from now. He has a collection of, I found a collection of about 700 specimens collected within like 3-4 years in Manaus. And as much as I know, TI is the only place where we have those plants, that collection and that record of that expedition. So these are some examples of what we did, but just to show you how the situation in TI Herbarium is now regarding like how data is open for people to see. if I take out data from GBIF, for example, data that is already available for people to search in GBIF regarding tracheophytes, vascular plants in Japan, these are the three main hits that we get: the National Museum of Nature and Science, TNS, the Kanagawa Prefecture Museum of Nature and Natural History, and the Nagano Environmental Conservation Research Institute. TI is not one of the highest scores. TI actually accounts for very few numbers, and if we compare to the total number of specimens that we have in the museum, that's about 2.2% of the collection only, within ferns close to 0%. So what that results is that data is not available, either like in Japan or outside of Japan. That's not only critical for us to think on conservation strategies to the collection, but also it limits how people has access for research and education using those things, particularly for those samples that are of plants that are not from Japan originally, that were collected out of Japan. So it's kind of an urgent mission for the TI Herbarium, for the University of Tokyo, to work on in my opinion, work on the databasing and making that data available to other people. So we are, of course, working with the digitalization databasing. The work is mostly done by myself and just one or two other assistants. So we don't have, if you know herbaria here in Japan, we don't have much space, and human resources are usually very restricted, as other places outside as well. But I don't know, I find Japan a bit like of a critical state already. But we are working still on the digitalization. We have reached close to 7,000 specimens. Most of them are available on the website of the herbarium, and we are not progressing towards making it available in GBIF and also in other sources like the National Museum of Nature and Science, the Science Museum Net, SNet, which is a national online database that we have. At the same time, I'm also working on non-digital forms of databasing. So we are constructing this catalog of the type specimens of ferns and lycophytes of the museum. It's going to be a 3 volumes catalog. The first one is done, it's in press, includes 16 families, and it's going to include all of the type specimens that we have found with some annotations and notes regarding groups that need lectopification and things like that. So I don't know, I came, again, not as a historian, but like I tried to bring a bit of the history just to maybe highlight the value of this collection in an international scope. And basically, these are the conclusions that I have. The University of Tokyo collection includes specimens that are important not only for Japan, but also for outside of Japan, particularly Taiwan and Korea collections. And databasing is not done. We don't have access to the information that needs to be accessed as soon as possible. And that needs to be, in my opinion, done while we have inter-institutional collaborations, not only nationally, but also internationally. So I don't know, that was my mission coming here today to talk with you, to bring this message. I wonder if this is enough for an opening for this session, actually. But I'm really looking forward to what kind of feedback I get from you guys. And thank you for your time. Okay, sorry, thank you guys.
Okay, our next speaker is Tokei Alberts, uncovering a history of our natural history collections, an early career experience digitizing California museum specimens.
Hello, สวัสดีครับ คนชื่อโตเกะค่ะ My name is Tokei Alberts, and I am a wildlife conservation post-bachelor's and a museum-based researcher. I am here representing 3 Californian institutions. The Cal Poly Humboldt Vertebrate Museum, the California Academy of Sciences, and the Museum of Vertebrate Zoology. I was inspired to give this talk after becoming involved with the Ranges Project, which is a 4-year collaborative grant to digitize and publish over 1 million traits from Western North American museum specimens. Specimens exist not only as representatives of their environment, but the products of scientists who deem them worthwhile for research. And the history and context surrounding each contributor, and by extension each collection, inform how they can stay relevant now and a century into the future. In my talk, I will be covering a brief history of the natural history collection field, some of the specific histories of my institutions, and the specific work that I've done through Ranges. As you may know, natural history collections originated as cabinets of curiosities, where specimens from archaeology, geology, art, and biology would be displayed all right next to each other. These biological specimens were collected by the first naturalists, often people who were independently wealthy. In the 18th century, we systematized our understanding of biology and refined our understanding of the natural world. And we also improved our methods for studying animals and the methods to better preserve and store the specimens that we are collecting. In the 1800s, mammal specimens were prepared more like taxidermy, with the skull inside of the skin. Following Darwin's publication of The Origin of the Species, it made more sense to store the skull outside of the skin in order to better study variation across species and geographical areas. In 1846, Spencer Baird, future Secretary of the Smithsonian, developed the first modern guide for the preparation of mammal skins and their tagging system. This was further refined in 1889 by C. Hart Merriam when he published a detailed and illustrated guide denoting the methods to better preserve specimens and record their external measurements, which are now preparation standard. Up until the 1970s, this also included the frequent use of arsenic soap as an insecticide to be brushed on the inside of the skin. Make no mistake, they knew it was bad. When a student of Merriam's reported suffering from arsenic poisoning, symptoms included green fingernails and open sores on the hands. Merriam admitted that he too had also suffered poisoning in the past and just recommended using a brush to clean underneath your nails after preparing specimens. In fact, the guide that I learned to prepare study skins on was published in 1962 and also recommended the use of arsenic soap. Despite these occupational hazards, we can study specimens in a hypothetically unlimited number of ways as we continue to revisit and interpret their data. In this way, we can build upon the legacy of researchers who have come before this, continuing the work that they did not know that they were participating in. In 1853, 3 years after California became a state, the California Academy of Sciences was founded in San Francisco. With San Francisco becoming an economic and shipping hub of the coast, it wasn't long before researchers embarked on collecting ships both within the state and across the Pacific Ocean. Unfortunately, CAS was devastated by the 1906 earthquake and fire, and it suffered a near total loss of their collections, save for the heroic efforts of their herbarium curator, Alice Eastwood, who scaled the burning building to retrieve irreplaceable herbarium-type specimens. For other departments, this history has directly shaped the Academy's collection, whose pre-earthquake Bay Area mammals are made up entirely from private collectors or for local universities. I came across a native rabbit specimen from San Francisco that was purchased at a meat market and prepared for someone's personal collection. My time on Ranges at CAS has particularly been spent addressing the challenges of using their database system, Specify 6. The way that they have historically been using it is not entirely conducive to the goals of Ranges, namely that trait data is concatenated to a single field, which makes it difficult to access and update on a large scale. This also limits the support that we can receive from Ranges and the knowledge that we can help transfer to other institutions. Going forward, we'll have to take some time to understand how to mitigate these issues and see how best to address this database approach. So the MVZ was founded in 1907. The MVZ is the Museum of Vertebrate Zoology by Annie Alexander. She was a sugarcane plantation heiress and funded biological research from her immense family wealth. She supported students and staff in order to create one of the first repositories of California wildlife. Over the course of her life, she donated tens of thousands of specimens and funded the men who would become the founding fathers of modern vertebrate zoology. At the time, field and research work were viewed as separate occupations, but she insisted that the staff at the MVZ split their time between research, teaching, and fieldwork, which the staff still continue to this day. She funded collecting trips for graduate students. For one of the most notable ones is Joseph Dixon, who was a very famous mammologist and ornithologist. In 1913, he embarked on a collecting trip to Siberia and Alaska, where his boat was stranded in ice sheets off the coast of Alaska. And he survived by hunting seals and seabirds, some of which might have otherwise become specimens. In fact, unbeknownst to him, while he was collecting semi-palmated sandpipers and redneck phalaropes, World War I had begun across the world with the assassination of Archduke Franz Ferdinand. In fact, he returned in October 1914, over a year late to his own wedding. Annie propelled the MVZ as a research institution with the hiring of their director, Joseph Grinnell. Grinnell viewed specimens as part of their ecology and really emphasized the collecting of multiple individuals from different environments, which directly led to the description of distinct subspecies. He also taught the natural history of vertebrates class, which is still taught today, and trained generations of naturalists. He is best known for his legacy of a very in-depth, thorough, systematized study of specimens, not just for mammals, but birds and herps as well, all across California and all of these different ecosystems. And he kept the most incredible notes. And it's because of these notes that in the late 2010s, the MVZ was able to resurvey these transects and replicate them and study how these species had changed and adapted due to climate change. This wealth of data has been published on already, but the vast majority of the mammal collection at the MVZ, including the original specimens for the Grinnell Survey Project and the Resurvey Project, do not have their trait data published and accessible on the database system Arctos. We are prioritizing them as their external data measurements makes them excellent candidates for climate change research. Along with the MVZ mammal curator, Chris Conroy, I have spearheaded the Ranges project, and we are making fantastic progress in bulk processing and uploading the specimens. I am in charge of generating, standardizing, and uploading the sheets, meaning that I have full control over the data from beginning to end, which significantly streamlines our digitization workflow. In the first 1 year of Ranges, we have processed 25,000 specimens and resulting in 100,000 traits. And this is all shrews and one species of deer mouse. This represents 13% of our collection. So this will be an incredibly large and valuable data set when the Ranges project is complete. But for the Paramyscus maniculatus deer mice and the shrew data, it is uploaded and accessible on Arctos for anybody to use. I attended Cal Poly Humboldt in my undergraduate, and at Humboldt they have a vertebrate museum. In 1969, Humboldt professor Tim Lawler kickstarted the expansion of this collection and over the course of his career increased the number of specimens tenfold. His philosophy of student involvement in the Vertebrate Museum continues to support students by giving them research projects and hands-on opportunities. Vertebrate Museum alumni have become pillars of the collection and mammalogy community. They possess over 9,000 specimens in their research collection, and a very high percentage of these have been collected or prepared by students either on collecting trips or in advanced fieldwork classes. This results in an incredible sampling of local redwood forest biodiversity. And while I was there, the Vertebrate Museum migrated from Microsoft Access to the Arctos database system, and we needed to identify some of their collectors to properly link and credit them. To do this, I read field notes, class rosters, and online obituaries, and uncovered that some of the museum's strongest relationships were forged for life. I was also learning how to make considerations regarding the maintenance of the museum and making decisions on its longevity and relevance. As we started with ranges, we needed to geo-reference their specimens and assign collecting localities and geographical coordinates to them. So in order to do this, I grouped, filtered, and used GPT-3 in order to standardize the localities and assign consistent formatting and abbreviations, primarily using geolocate and Google Maps. And when I was finished, I was left with a few thousand unique locations encompassing the entire research collection. And in a university collection like Humboldt, many specimens originate from very few specific localities. So you need a good understanding of some of the history of the alumni and some of these historical locations. Notably, there was a mountain beaver that was found outside of a bar, and I was able to ascertain the street address from an eBay listing for a matchbook. Also, very cool, there was a pocket gopher found here. This was posted in 1973 from an alumni of a student housing trailer park, which was described as a, quote, slum, and, quote, eventually unfit for human habitation. I also uncovered local industries like a whaling station, lumber mills, and drive-in movie theaters. So the data set of this results in showing the biodiversity of this small town university, but also the rich history of the people and places that support it. So 1 year into the 4 years ranges grant, I've been responsible for the data management of tens of thousands of specimens and gained a new appreciation for the ways we can learn from them. This data can be used to uncover not only how climate change and variance, but also how every museum and every specimen can continue painting the picture in which they lived. So I had more, but I would like to thank all of the institutions who have supported me, all of the people who have supported me, especially the California Academy of Sciences, Museum of Vertebrate Zoology, the Ranges Project, and the Spinach Travel Community. Thank you very much.
Thank you, Toke. Next up, we have Victoria Pickering, the Sloan Lab, mobilizing historical botanical data at the Natural History Museum in London.
At the Natural History Museum in London, the historical botanical collection was formed between the late 17th and mid-18th century, a context of enlightenment and imperial science and its social and political entanglements. It comprises of two parts. The first is the Sloan Herbarium, consisting of over 120,000 specimens arranged in 337 bound horti-siccae. This is a collection of collections. The second part is the Vegetable Substances collection, comprising over 8,000 surviving botanical samples sealed into small ornate boxes. Each box has a handwritten number, and this keys the contents of the individual boxes to a handwritten entry in a manuscript catalogue. This is the first page of that catalogue. It starts with the number 1, which you can see at the top, and continues on in numerical order. There are over 12,500 entries in this catalogue. And these descriptions really vary. At the very least, they describe what the object is. And from there, there's a real variation in detail about people, places, uses, cross-references. Both of these sub-collections were part of Hans Sloane's much wider and vast collection. Sloane, a physician, a Royal Society president, and an individual intimately bound up with the 17th and 18th century trading networks, had a catalogue process for all of these sub-collections. He hand-wrote descriptions of the objects across 40-plus catalogues, including fishes, serpents, and miscellaneous objects. Sloane gathered very few of these objects himself. They were amassed by a range of people from around the world. His correspondence is held at the British Library, which details many of these relationships and exchanges. When Sloane died in 1753, he bequeathed this vast collection to the British nation, and it went towards the founding of the British Museum, opening to the public in 1759. The natural history collections were moved to South Kensington in the 1880s, and thereby separating and dispersing objects that would have been considered together, text, image, and object. Researchers have long been attempting to unite these objects and their records, and recent digital attempts as part of Towards a National Collection, or TANC for short, are the latest iteration of this process. TANC is an AHRC initiative and a recent major 5-year investment in UK GLAM. The Sloane Lab is a discovery project. It is a multidisciplinary and cross-institutional project based across University College London, TU Darmstadt in Germany, the British Museum, and the Natural History Museum. It aims to facilitate digital access to the historical and present-day information that describes Sloane's collection, opening it up in more intuitive and relevant ways for public and academic audiences. Our focus at the Natural History Museum has been the historical botanical collection and making it digitally searchable and accessible for the first time. This is a collection formed, arranged, and documented before 1753, making it pre-Linnaean. The Sloane Herbarium is one of the largest surviving pre-Linnaean collections and is significant for a range of research. For example, it can make us think about processes of collecting and assembling and early encounters between cultures. This Opuntia specimen is from the Duchess of Beaufort's collection. She died in 1714. And she was responsible for introducing into Britain a wide array of garden plants. And this tea specimen was collected in Japan by Engelbert Kempfer while working for the Dutch East India Company. As Sloane acquired these collections of herbaria, because it's a collection of collections, he and his assistants catalogued their contents using this 17th century publication called Historia Plantarum by John Ray. This is a 3-volume work in Latin published between 1686 and 1704. It aimed to create a worldwide encyclopedia of the flora then known to European natural historians. The publication is also pre-Linnaean. The plant names it itemizes are found in a polynomial structure, creating long plant names in Latin which act as descriptions rather than identifying labels. And it's important to note this because today a binomial name rather than a phrase name is used to classify plant material. This is Sloane's personal copy that we're talking about, and in the side margins of the folios, there are thousands of handwritten annotations next to printed plant names. You can just say, yeah. These annotations take the form of a volume number and a folio number, thereby acting as an indexing reference to the physical location of a specimen within the herbarium. So you can look at an example here, HS, so Hortus siccus, not Hans Sloane, volume 85, page 86. So that's saying there's a specimen of this plant on this page in this volume and on this page in that volume. At the top and bottom of the pages, we find additional handwritten annotations which contain plant names and specimen references following the same format. And here it's a case of the plant species names are those not already included within Ray's list. So this paper tool is the primary way to find specimens in the NHM's founding botanical collection. And until now, a user has had to be physically located in this room where the collection is and where this text is to be able to physically access and search for a specimen. A user must also hold botanical and historical expertise to navigate between the polynomial plant name descriptions found in Ray and binomial nomenclature of today. So to extract these plant names and connect them with the physical location of the specimens would unlock this collection in so many ways. So what did we do as part of the Sloan Lab? Well, so we used high-resolution scanned images of all 3 volumes, and we had 3 workflows. We focused on the printed plant names in the middle of the text, the handwritten annotations in the margins, and then the headers and footers. A key part of this entire process was the need for those handwritten annotations to be correct, because or as accurate as possible, because if they were not accurate, it becomes a useless resource, because if we get the page number wrong or the volume number wrong, we can't find the specimen. Our digital humanities colleagues used rule-based algorithms to create boxes around the information we needed based on the structure of the page. Then the contents of those boxes were automatically transcribed using OCR and HTR techniques. And then we captured the relationship between the plant name and the corresponding specimen, because that's really crucial. During our testing phase, it became clear that it would be difficult to extract the information accurately. We received the data and recognized the volume of errors was really significant, and we realized that we would need to check everything. Every bit of data that came our way, we had to check. And so this had a huge impact on our workflow. We really had to ask ourselves, what is the essential information that we require within this text? And we had to remind ourselves that we were not looking to complete a document analysis of this text, but we were looking to create a tool for accessing the collection. So this meant rather than capture every single plant name within the text, we only captured those that had specimens associated with them. So we extracted the plant names and we checked them. We used Zooniverse to transcribe the numbers, and we checked that output. And then we outsourced the transcription of the headers and footers to an external company, and then we also checked that output too. So across all 3 volumes, we've extracted and checked over 9,000 plant names, but that doesn't include the headers and footers of volume 3, that's still coming in. Across volumes 1 and 2, we've extracted over 27,000 specimen references, and as of last week, we can add 13,000 specimens from volume 3. So that's almost 40,000 specimens across Ray, which is about a third of the specimens in the herbarium, which is interesting. And all of this information is being uploaded to the Naturalist Museum's data portal as a data set. Despite its significant historical and cultural significance, though, we've not actually been able to answer basic questions about the Sloan Herbarium. For example, the number of specimens it contains or the geographical scope of the material. So we also completed an inventory of every photo to provide a framework for further data mobilization efforts. So this means we worked through every single folio in the Sloan Herbarium. And we recorded its contents and data associated with each folio. It took us over a year to work through all 337 bound volumes, which is almost 38,000 folios. And our best estimate, it's still an estimate, is that we have 121,505 specimens. As part of the Sloan Lab, we've also mobilized data relating to the vegetable substances collection. So there's a transcription of the catalogue on the data portal. That's a really rich resource that is searchable. And we also completed an inventory of the surviving boxes. So that means that we, again, counted the boxes, and we recorded 2 pieces of data, the object registration number, each item has a barcode on it, and also that original number on the boxes. So then what we were essentially doing is connecting a surviving box with original 18th century description. So now we can say that there are 8,815 boxes, and in a number of cases there are multiple catalogue numbers per box, which makes us think about what was a specimen for Sloan. So as a result of the Sloan Lab project, and this is the whole project team here, we have mobilised over 40,000 data points for the Sloan Herbarium and 20,000 for the vegetable substances. And we've learned a lot from this process. There's much work still to be done, but the data is allowing for new questions to be explored and a framework for data enrichment, focusing particularly on people and places to facilitate searching the collection. But we've been able to begin making comparisons across these sub-collections for the first time and really start to consider some broader questions about 18th century botanical collecting and collections management. Thank you.
We have time for one question. Whoever's fastest.
Are you sharing your presentation? And I know you said Zooniverse, but that's actually people who are transcribing those records. Can you share your presentation with them? Because I know as a transcriber, when I volunteered, I'd want to see that. I'd want to see the outputs of the transcription.
Yes, and also, so the data set that we've put up on the NHM portal, we have listed, there was 6 individuals who helped us transcribe, and they're all authors. That was a really important part of it. Yes, no, that was a really important part of the work, was acknowledging those contributions. So, so, and they have access to that data set, of course, and the references. So,
Cool, thank you.
We have time for one more question if it's very short.
As I do, sorry, I'm Nicole, Biodiversity Heritage Library Australia. I was just desperately searching for it on BHL. I think I've found it. Is it, if it is the one that's up on BHL, which was uploaded in 2017, then that's no?
The copy of John Ray's Historia Plantarum, you mean?
No, Sloane's Herbarium Annotated is on BHL, uploaded by NHM in 2017.
Yeah.
I'm just wondering if that's it that you've been talking about?
So that's Dandy's Guide to the Sloane Herbarium, published in 1958. Yes, which we uploaded to BHL, which is an amazing resource and is one of the really only sort of descriptions and guides to the herbarium. And we have a PhD student called Brad Scott who is doing a PhD on the herbarium and who has used TEI and XML to encode that copy of Dandy and basically pull it apart and find out what is missing, because it's missing a lot of information, but it provides really great context. So that is also, so Brad's data has also been uploaded to the NHM data portal. And what we're hoping for is that all of this is going to go into the Sloan Lab. So it's not just a quite particular list of volumes and specimens, but there's that kind of contextual information that's coming out of Dandy, because Dandy has descriptions of people and places, but it's patchy, which is what we're learning from the process. Does that answer your question?
So my other question was, can you put it on BHL if it's not there already?
The
actual, you've digitized the entire thing, so.
Yes, the data, so just to clarify what we mean by digitized in terms of images or the information about the volumes, because we only have about 3,000 images of the 121,000. So that is part of this process, which is we've essentially created a framework for the data, sort of to hang more data on. So at the moment, what we have produced is a list of volumes and the folios, and now we've got those plant names from Ray, so we know what they are, and it's really the basis that we can add to. But it's a good question about what part of that we would put up on BHL.
Yeah, well, thank you.
Thank you.
Okay, our final speaker today is Gary Aronson with A Browse Apes, Historical, Cultural, and Scientific Structures Surrounding an Orangutan, Pongo Spa, collection at the Yale Peabody Museum.
Thank you. Hi. I want to thank the sponsors of this program. I want to thank the organizers, and I want to thank the amazing audiovisual team for everything they've done. 2 announcements before we start. Number 26, your food is ready. 26. I'm never taking this off. And number 2, I am an interloper here. So in the words of a famous 20th century poet, I am a freak. I am a weirdo. What the hell am I doing here? I don't belong here. But I do want to present on work I've done with several of my students and with El Presidente himself, Greg. And so this is work that we've been doing at the Yale Peabody Museum, established in the 19th century, with a relatively large vertebrate zoology collection. And within that collection is a series of primates. In 2016, I organized a detailed review of the ape collection that is present, where we want to go through every single set of cataloged elements and then provide you with detailed information about taxonomy, life history, and other variables relevant in understanding aspects of primate and ape biology. You can find the publications of the work that we've done thus far in the Yale Peabody Museum Bulletin, and I strongly encourage you to review these pieces of work, as the students did an amazing job. Today I'm going to be focusing on one specific project, and that project is the work we've done on the orangutan collection. I am struggling to follow such an amazing presentation where we're talking about tens of thousands of things. We're going to be talking about a very small subset of individuals here. Peabody Museum's collection contains 9 orangutans, relatively small in some respects. However, the collection does contain multiple age classes. We have both wild and captive individuals represented, and there is some taxonomic diversity. And so, again, the student and myself had worked through these particular individuals to try and provide the best description possible for anybody interested in primate anatomy and biology to visit the collections at the Peabody Museum. Among one of the findings that we had is that 4 individuals from the Yale Peabody Museum's collection have the accession and provenience date of coming from the Yale School of Medicine in 1932. At the last check for us, we did not realize that there were endemic orangutans in New Haven, Connecticut, so therefore this led to the question, how did this come to be? And in order to understand this, we begin to delve into these questions of the history of science and collections. In the 1920s, the Yale president and the deans of the law and medicine schools had decided they wanted to organize a program with the Harvard psychobiologist and primate ecologist, or primate behaviorist, Robert Yerkes. And in 1929, they had organized an Institute of Human Relations, which ostensibly was kind of a multidisciplinary program where they wanted to synthesize science, policy, and other practices and measures to try and develop a kind of a multidisciplinary center that Yale would be the head of to try and inform aspects of the human condition. And the graphic you see down there with the little star just basically shows you how this institute, and that is still the same facade of the Yale School of Medicine today, how this institute was integrated into these larger programs. I want to be sure to point out that both Angel and Yerkes were prominent supporters and implementers of the theories of eugenics, which is both racist and wrong, and the implications of their work continue to reverberate through the United States of America today. The Institute had formed multiple laboratories, and within these laboratories was a strong emphasis on primate neurobiology and comparative anatomy, to the point that they were using these primates to try and understand more about the nature and structure of the evolutionary biology of the brain and of behavioral science. And so it was clear that models needed to be built to organize questions about the anatomy and the structure of brain function and innervation. But to do so, you need live primates, which led to the question, how do you propagate, manage, and continue to build a colony that you can use as study subjects? Here, we introduce Rosalia Abreu, who is a Cuban national who is part of the aristocratic families that were present in this part of Cuba at the time. She has a remarkable history, and her family has an enormous impact on many aspects of Cuban history. She moved to Paris. She had married a physician. She was divorced, and then in the 1900s, she returned to Havana and moved into basically the Quinta Palatino, which was a very large estate located in Cuba. One of the reasons it's important to understand who this person is because Abro's work is related to her fascination with exotic animals. In fact, her work had brought her from France, and then she actually started purchasing apes, monkeys, and a wide array of different kinds of animals. And so in Havana, in this massive estate, she built a large compound with multiple species, cages, and enclosures. And by the time we get to the 1920s, she has about 60 primates on the scene, including 3 orangutans and 14 chimpanzees. That's a lot of animals to have in one place. Most importantly, and the reason why I want to make sure you understand her contribution is that she is the first to have successfully managed to breed captive apes. Multiple attempts have been done across the world to try and keep these animals alive with 0 success. So her skill in managing these animals in this captive environment led her to be quite famous among the peers at that time. And Yerkes knew this. Yerkes was in correspondence with Abro early in his career, and he was invited by her to come and visit Cuba with the hopes that she would be able to establish some kind of a relationship with Yale University, with the United States, to provide this kind of, this base of study subjects. However, Yerkes' plan was to make sure that he established a primate center at Yale, and so again, using funding from the Rockefeller Foundation, he had managed to organize a primate center at Yale University, and this concrete or brick barn is just down the block from my office, and that is where captive chimpanzees were housed at Yale University for the first time. They eventually did build and become part of the Yale School of Medicine, a much larger facility. Yerkes began working with chimpanzees and monkeys and doing his research there. For those that are familiar in the United States, the Yerkes program moved down to Florida and then eventually and is still present in Atlanta, Georgia today. Now, in 1930, Rosalia Abro passes away, and her will is very explicit. No matter what, she does not want any medical experimentation or intrusive procedures done on any of these animals. Her son does not have this perspective. Her son writes to Robert Yerkes and says, you can select whichever animals you like, because his plan was to remove and disband this menagerie. There were negotiations between the Institute of Human Relations and Pierre, the son, and there were letters and correspondence that are present indicating that Rosalia's will make it clear that no invasive procedures are to be done. There is an agreement that 4 orangutans are going to be moved from Cuba to Yale for a cost, today would be about $42,000, 2 adult males and 2 adult females. Just as a brief side note on this point, in 1931, 1932, all of the orangutans were transferred to Cuba, and the Philadelphia Zoo in the United States is among the oldest zoological gardens that we have, and 2 of the orangutans from Abro went there, and they are actually among the longest-lived captive orangutans in history. And so their skeletons are currently at the Smithsonian Institution. I have not seen them, but it's important to remember, as you'll see soon, that her success in bringing these animals into this other institution. On 11 July 1932, Yale University receives 2 females, relatively young, and each of these females had a baby sired by that adult male you can see up there. Thanks to the work of one of my co-authors, Catherine Moss, and working with the archives at the Yale School of Medicine, the detailed records of each one of these animals is present, as any good researcher or scientist should have. There's a lab notebook providing details on each one of these animals. And so here we can see the individual Darling, one of the females that was present. Here we can see Cachita, the relatively larger female, and it's noted multiple times in these notebooks that she has immense strength. This is Cachita's son, Mim, and this is Darling's son, Carlito. And so again, these records provide relatively detailed information about each one of the study subjects. And these notebooks are present for any of the primate subjects that were conducted at the Institute of Human Relations. I sincerely regret to tell you that in one slide I can give you the fate of the 4 orangutans. When they arrived, both of the babies had come in with helminth infections, and they subsequently developed respiratory infections relatively quickly. They were treated with whatever methods were available. There was every effort made to make sure that they could prolong and keep these animals alive. Regrettably, they both died within days of each other, within 1 month of arrival. The larger, immensely powerful Cachita bent the bars of the cages that they were placed in the Yale School of Medicine, requiring reinforcement. On the evening of 25 July 1932, she dismantled a wall and she pulled a lantern gas pipe out, and I regret to say that she had passed away through asphyxiation. Darling was, in fact, subjected to a surgical procedure, left foot extirpation, which is basically severing nerves within the brain to injure and alter function to mimic aspects of stroke or other aspects of neurological disorder. She developed sepsis as a result of this operation and died in a relatively short amount of time. Again, these animals arrived at considerable expense and considerable effort by all parties involved, and within 1 month, all 4 of these animals were gone. And I remind you again, the Philadelphia Zoo has the longest surviving orangutans from the Abreu estate, and this was another impetus to make sure that the Floridian compound, to make sure that these captive apes could be managed more effectively, had to be an essential requirement because the New Haven operation was not working. Following the necropsies and the detailed analyses, again present in all of these medical notebooks, the director and the mammalogy curator were contacted, and the skeletons and skins are present in this collection today. There were publications that came out from this work regarding nature of the physiology and anatomy of these different brains of these and other primates. And one of the researchers, Paul Busey, his work is still relevant in some aspects of medicine in terms of strokes and dyskinesia. I have not found any other information about any of the studies or samples that were removed from these particular individuals at this time. Upon Rosalia's death, the entirety of the menagerie of an amazing array of wild animals were distributed all over the place, and the property was abandoned within Cuba for some time. Today, it has been refurbished and turned into the Technological Palace of Cuba, and is also a well-known tourist center. And it is, her family, and this place is quite famous within Cuba. And so everybody knows about the amazing animals that were there at some time. Rosalia Abreu herself recognized the nature of how science worked both in the 20th century and unfortunately some aspects of the 21st century today, that she wrote in her letters that she has made massive contributions to science, but she still needs a scientific man to work with. I want to make it abundantly clear that without Abreu, there would not be a Robert Yerkes. You may not know who this is, but this is among one of the most famous primate ethologists in the United States of America. Without Abreu, there would not be American and international primatology from both my perspective, professionally and personally. And without her education, Yerkes would not have been able to develop, as you can see in the publications over there, his amazing array of successes in chimpanzee and ape husbandry within the United States. I do want to be very clear, Yerkes always made sure to give her appropriate credit, so it's not like she was forgotten from that perspective. But Rosalia Abreu is not as well known as Yerkes or these other researchers that are famous in their fields of neuroanatomy and biology. I reiterate that this case is telling us very much about the nature of how anatomical work is an issue, and that invasive ape research is no longer supported within the United States. Simply put, this study is an example of how it is that we can use studies like this to understand aspects of primate biology, evolution, and conservation, but these collections also illuminate historical events, biases, and other issues. The Peabody Museum's records, while very valuable and useful, did not have these details, and so we are working hard to make sure we can update those. And in fact, the Pongo Orangutan Stud Book, which represents all captive orangutans, the individuals that I'm talking about here don't exist in that, so I need to find out how to work with the American Zoological Association to put them in. These individuals now have very detailed life histories as well as medical journals associated with them. And again, this work, working with colleagues like yourselves, working with natural history communities and researchers and staff, provide us with a much larger perspective on the historical as well as the biological nature of our collections. I do not think I have any time, and I apologize for that. If you do have any questions, you can find me at the Elegance Karaoke Pub tonight, because it's been a dream to come to Japan, and I really appreciate the opportunity to be here. Thank you very much.
All right, thank you, everyone. That concludes our session for this afternoon. If you're in person, please go and have some tea, which is actually coffee. If you're online, our next sessions start, I believe, at 4 o'clock.